Welcome to the Deep Dive, where we translate the cutting edge of scientific literature into essential knowledge. Today, we're taking a deep dive into long read sequencing, a technology that's, well, truly transforming the field of cytogenomics. For you, a highly skilled specialist in this area, our mission is to distill the most impactful advancements, their applications, and the real world challenges as this field rapidly evolves.
Exactly. And what's truly revolutionary here is how long-read sequencing, or LRS, is fundamentally altering our understanding of the human genome. It's directly addressing these, long-standing barriers that conventional methods simply couldn't overcome. We're going to explore how technologies, particularly from Pacific Biosciences and Oxford Nanopore Technologies, are dramatically enhancing capabilities crucial to cytogenomics, everything from resolving complex structural variants to achieving complete chromosome-scale assemblies.
Okay, let's unpack the problem LRS emerged to solve first. Because for years, we've relied heavily on short read sequencing, SRS, and also chromosomal microarrays, CMA, in genetic diagnostics. They're cost effective, sure, and accurate for single nucleotide variants and smaller changes. But for all their utility, they've consistently left us with pretty significant blind spots, especially in areas central to your work in cytogenomic analysis.
Yeah, if we zoom out a bit, the core limitation really stems from the short read lengths, typically just 75 to maybe 250 base pairs. This means they simply cannot unambiguously map to the large repetitive regions that make up, well, over half of our human genome. Think about it. Elements like short tandem repeats, lines, semis, segmental duplications. It's a huge chunk. For instance, thousands of exons across hundreds of genes regions, absolutely critical for diagnosis, remain effectively inaccessible or highly challenging for SRS. This often leads to fragmented assemblies and an inability to resolve structural variants larger than, say, 500 base pairs for SRS or maybe up to 50 kilobases for CMA. Essentially, we're missing crucial information about their precise location and orientation.
So, for you, the specialist, what does this ultimately mean for diagnostic yield? I mean, for patients, this often translates into that prolonged, incredibly frustrating diagnostic odyssey, right? Requiring a battery of different tests. Consider, for example, a comprehensive hearing loss panel that might demand a complex, tiered approach combining multiple techniques. Even then, inversions or really complex rearrangements in challenging genes like STRC or ESPN can still just elude detection. And when dealing with autosomal recessive conditions, accurately phasing variants often hinges on sequencing parental samples, which, let's face it, isn't always practically feasible for families. This leaves a significant proportion of individuals without a definitive molecular diagnosis. That's really the gap we're talking about filling. Okay, so here's where it gets truly transformative. Long read sequencing emerged specifically to tackle these fundamental limitations. We're talking reads that are orders of magnitude longer routinely from 10 kilobases up to 100 kilobases, with a current record shattering at, 2.3 megabase. This isn't just a quantitative improvement, it's a qualitative leap. It's profoundly reshaping cytogenomics. It's like going from, I don't know, black and white outlines to a full color 3D scan of the genome, revealing all the hidden details.
Precisely. That's a good analogy. The real game changer is LRS's ability to span these complex genomic regions. It provides unprecedented resolution and delivers answers where previously we only had sort of fragmented clues. So first, its superior resolution of complex regions. This means LRS can directly sequence through extensive repetitive elements, including those previously considered, well, intractable, like centromeres and telomeres. This capability has been crucial in closing gaps in existing reference assemblies, even enabling the first complete telomere-to-telomere assemblies of entire human chromosomes, like the X, and ultimately the entire human genome. Secondly, for comprehensive structural variant detection, LRS is uniquely suited to identify large structural variants, SVs like insertions, deletions, inversions, that are routinely missed by short reads. This includes those notorious repeat expansions that define many neurological disorders. Think Fragile X syndrome or the hexameric repeat in C9 or 72 for ALS. It can even resolve complex things like polyalanine tract duplications in genes like HOXD13.
That's incredible, particularly for conditions like Fragile X, where that repeat expansion is the core pathology. So, you're saying LRS can essentially quantify and even phase these expansions with a precision we just couldn't get before, often within the same patient sample.
Exactly. It's not just detecting them, it's characterizing them with the specificity needed for clinical action, allowing us to differentiate, for example, between a full mutation and a pre-mutation allele, maybe within a single read, which leads right into its third major advantage: accurate long-range haplotype phasing. LRS infers phase with high accuracy over long genomic intervals, sometimes spanning entire genes. This is absolutely critical for definitive diagnoses in autosomal recessive conditions, determining if two variants are on the same chromosome, cis, or different chromosomes, trans. It often reduces the need for those tricky parental samples. We've seen this directly in cases where LRS resolved inconclusive diagnoses for hearing loss, differentiating between variants in TRAOBP that were cis and complex findings in STRC involving a gene conversion event that turned out to be trans.
Wait, okay, so you're saying LRS can directly detect epigenetic modifications without bisulfite conversion. That sounds like a huge workflow simplification and potentially eliminates artifacts too. Can you elaborate on the diagnostic significance of that? Maybe another example beyond Fragile X?
That's its fourth key strength, direct epigenetic modification detection. Yeah, LRS platforms can directly detect chemical nucleotide modifications like 5-methylcytosine and 5-hydroxymethylcytosine without needing bisulfite or PCR. This is diagnostically crucial, as you mentioned, for understanding methylation patterns like the hypermethylation of CG repeats in Fragile X, which has direct clinical implications. And finally, for RNA sequencing, its fifth advantage is full-length isoform resolution. LRS can sequence the entire length of a transcript in a single read. This leads to discovery of novel reading frames, isoforms, allele-specific expression, variations in post-transcriptional RNA modifications. It offers a much more complete picture than trying to computationally piece things together from short reads.
Right. Okay, we've talked about the incredible what LRS can do. Now let's pull back the curtain a bit and understand the how. What's the fundamental magic behind PacBio and Oxford Nanopore that allows for these revolutionary long reads? How do they work?
Okay, let's start with PacBio. Their single-molecule real-time, or SMRT sequencing, especially with its high-fidelity hi-fi reads, offers exceptional per-base accuracy. We're talking exceeding 99.9%. Hi-fi reads are generated by having the DNA polymerase do multiple passes on a circularized template. They call it SMRT-Bell. A consensus sequence is derived from these multiple subreads. Their new Revia system is a really significant leap. It increases hi-fi throughput by 15-fold, enabling something like 360 gigabases of hi-fi data per day, and it completes a full run in just 24 hours. Crucially, this also comes with a substantial reduction in cost per gigabase. So for your lab, this means moving from maybe niche research applications towards potentially high-volume diagnostic throughput. It makes the technology much more accessible for routine use.
That's a really impressive leap for PacBio's accuracy and throughput. But PacBio isn't the only player defining this space. Oxford Nanopore offers a distinctly different, equally powerful approach, doesn't it?
Indeed. ONT platforms like the Portable Minion, the Gridion, and the high-throughput Prometheon, they're renowned for their ability to generate ultra-long reads, routinely tens to hundreds of kilobases, and sometimes even several megabases in length. They often outperform PacBio in just raw read length. Now, while early ONT reads had higher error rates, recent advancements with their Q20 plus chemistry and the ligation sequencing kit V14 have pushed accuracy way up over 99%. A truly unique advantage is their real-time, direct electronic sequencing. As DNA passes through a nanopore, changes in electric current are directly translated into sequence. This allows for immediate analysis. And as we mentioned earlier, this also enables direct detection of modified bases through characteristic signal shifts without any chemical conversion needed.
Okay, so beyond just the raw sequencing, LRS has dramatically advanced the complex art of de novo genome assembly. For years, piecing together a genome felt like, well, you use the analogy, assembling a massive jigsaw puzzle without the box cover, and often with thousands of pieces from entirely different puzzles. With long reads, it feels like we finally get the box cover, or at least much bigger chunks of the border.
That jigsaw puzzle analogy is spot on. Long reads directly address the core challenges of de novo assembly, which is especially critical in highly heterozygous or polyploid genomes. By spanning those long repetitive regions, LRS helps us avoid biases from reference mapping and construct far more contiguous sequences. Key assembly algorithms have been refined for long-read data. Tools like Canoe and Hyphiasm use an overlap layout consensus, or OLC, approach. That essentially builds contigs by finding overlapping sequence ends, while other tools like Shasta and Fly use De Bruijn graph, DBG approaches, which break reads into smaller kernels and connect them, sort of like building a map.
But the real power often lies in combining strengths, doesn't it? Not just one approach.
Precisely. Hybrid assembly, which takes the high accuracy of short reads and combines it with the long-range information of LRS, offers an incredibly powerful strategy for highly accurate and contiguous genome reconstructions. Furthermore, integrating Hi-C data, which essentially tells us which parts of the genome are physically close to each other in the nucleus, even if they're far apart linearly, is crucial for achieving true chromosome-scale contiguity. and for generating phase assemblies. This was instrumental in the Telomere to Telomere Consortium's complete human genome assembly. It finally resolved those previously unsequenced regions like centromeres and the acrocentric short arms.
Right, the TTT assembly was a landmark, and ensuring the quality of these complex assemblies is absolutely paramount in a diagnostic setting. How do you actually validate something so intricate?
That's a critical question. We assess quality along three vital dimensions. There's contiguity, often measured by metrics like N50, which tells us the length of the shortest contig at which half the genome is covered. Then there's completeness. We often evaluate that using tools like BuSCO to check for expected conserved genes, or maybe Mercury to look for missing kamers. And finally, correctness. This verifies the accuracy of each base pair and the overall structural integrity of the assembly using tools like QUAST. Beyond these metrics, the evolution of error correction and polishing tools like Medica and HomoPolish also plays a vital role. They refine draft assemblies, rectify systematic errors, and improve the overall consensus accuracy, even with the high inherent accuracy of Hi-Fi and Q20 plus reads.
Okay, so as exciting as these advancements are, let's be pragmatic for a moment. For you, the cytogenomic specialist wanting to integrate LRS into your lab today, what's the single biggest practical hurdle you're likely to hit first? Is it sample quality, data analysis, maybe cost? And how might you start to address that?
That's a really critical point. While cost and throughput are evolving rapidly, for many, the biggest initial hurdle is often the sample preparation. Specifically, getting that high-quality, high molecular weight DNA needed for optimal LRS. Routine clinical samples like saliva, for instance, they're convenient but often just fall short. You frequently need blood or perhaps other more invasive tissue samples to get the quality you need. And while cost, though definitely decreasing with systems like PacBio Revio, it still remains higher than short-read whole genome sequencing. That makes it less accessible for routine, high-volume diagnostics unless you use targeted enrichment strategies. Throughput is also improving dramatically, with Prometheon offering ultra-rapid sequencing, maybe under a day for critical care settings, but it still needs to scale further for truly population-level studies.
And what about the computational side for you, the specialist working at the bench? That's a huge piece of the puzzle too, right? The
Yeah.
bioinformatics.
Absolutely. For widespread adoption, we really need highly intuitive bioinformatics platforms that seamlessly integrate into existing clinical workflows. Along with that, we need revised regulatory accreditation and credentialing frameworks to catch up, automated assembly and phasing of deployed human genomes at scale. Well, that remains an area of very active development, pushing the boundaries of what's possible for high-volume clinical applications.
Okay. Despite these challenges, the overall vision is incredibly compelling.
Indeed, the future promises things like the Pangenome Project, aiming to create a comprehensive collection of diverse human diploid reference genomes using LRS. This will be transformative for personalized medicine, moving us beyond just a single linear reference genome. The continuous innovation in LRS technology, driving down costs and improving accuracy, is clearly accelerating its integration into clinical settings. It's happening fast.
This deep dive has really underscored how long-read sequencing isn't just an incremental improvement. It's a truly fundamental shift in our ability to decode the human genome. It's empowering you, the cytogenomics specialist, to resolve previously intractable regions, identify complex structural variants, and accurately phase haplotypes. This fundamentally enhances molecular diagnostics and, crucially, can shorten those often agonizing diagnostic odysseys for patients.
Yeah, if we connect this to the bigger picture, the journey toward a truly comprehensive, personalized understanding of human genetics is definitely accelerating, and LRS is really at its core. The ability to reveal the full spectrum of genetic variation, including those previously hidden segments and complex epigenetic modifications, it promises to unlock new insights into disease mechanisms and ultimately pave the way for more precise and effective clinical interventions.
So for you, we leave you with this provocative thought. As long-read sequencing continues its rapid evolution, how will its increasing affordability, accuracy, and accessibility reshape the standard of care in your practice, moving us closer to a future where every genetic puzzle can finally be fully assembled and understood?